cable. I don't want.
This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
We're ready to roll? Okay, this is Paul Verschure with the Convergent Science Network, together with Tony Prescott. And our guest today is Yaki Setty, who was speaking at the BCBT Summer School on actually synthetically building different kinds of organs. So Yaki, how do we build a synthetic organ?
Well, first we have to know the biology.
Mm-hmm.
We have to know what God created or what biology created to rebuild it.
Mm-hmm.
We need to know the biology better than the biologist and better than the biology knows itself,
Mm-hmm.
because we need to reconstruct it. So in a way, I don't want to sound too sophisticated, but we're playing kind of creator.
Mm-hmm.
We create something. We use the computer as a platform to play with the data, put the data into the computer and play with it.
Mm-hmm.
And we know what the desired output is. We don't know all about it, but we know if something is missing, we will see it, that the output will be wrong.
Mm-hmm.
So that's the...
But no, this doesn't just happen by accident, right? It's not that you just have, let's say, a random set of ingredients like some big minestrone, and out of that comes an organ like a pancreas. So the first example you discussed was to really grow in silico a pancreas, and then you showed to us that model of the developed pancreas shared many features with a biological pancreas. So how do you So the pancreas is actually built from different kinds of cells, right? And the morphology of the structure will change over time in some developmental process. So how do we exactly model that? How do you model such a cell? And how do these cells then collectively form something that you could call a pancreas?
So in each cell, you put the biological data from the papers. So you have a block diagram, which blocks and arrows. And each arrow is a transit between the two states of the block. So you take the cell and you define the state it may be in. And this is correlated to the biology. Mm-hmm. You have to justify every block and every arrow you put
Mm-hmm.
in this diagram.
So for your cell model, how many states can the cell attain?
Well, it depends which model it is. The pancreas model, it's the most complicated. And it had over than 150 states all
Mm-hmm.
over. The number of the differentiation state was, I believe, like 10,
Mm-hmm.
which is more or less the number of markers that the biologists found. So
Mm-hmm.
for each state or stage, the biologists have defined we have a state in our differentiation component. And it's the same for other components in the system. For example, the proliferation component has 5 states, which it is correlated with the 5 stages of cell cycle. So we are trying to put into the model and to formulate as many biological data as possible and to be able to justify everything. For example, if we put an assumption or an error or a block in the diagram, so each one of this, we are able to say, okay, this data was taken from this paper.
Mm-hmm.
And that's why we constrain the model to be biologically plausible or accurate, or at least justified.
Mm-hmm. Right. But then, so we have the cell can have a large number of states. Then there are certain inputs to that cell that will make it actually decide which state it attains. Now, that transformation of inputs to the state expressed, that's more like a logical lookup table. Like if my inputs have the following characteristics, then in a deterministic fashion, I turn into the state 1. Or is that transfer function following more the kind of messenger systems that you would find within a cell?
Yeah, it is temporal decision. It's based on time and space.
Mm-hmm.
You will get some kind of message at a certain point if you are exactly at the certain position and has the same a specific message.
Mm-hmm.
So it is deterministic in that kind of perspective. If you look at the individual cell, But if you look at the population level, you will see that there are a lot of a lot of stochastic decisions are made. For example, if cell number 1 had made a decision at one point, and cell number 2 made a decision at another point, and they switch roles, so your simulation will be a little bit different. It is equivalent to two embryos, 2 human beings that are. All of us have 2 hands, have 2 eyes, have 1 head, 2 legs. But still, there is a lot of variety, and part of it is the genetic issue that we take with us, and part of it is part of our development. So this is why 2 brothers are not the same, and even identical twins are not identical.
Right.
So, I mean, your model of a cell is a kind of informational model where you describe the states it can be in, the kind of switches that within it, whether the switches can be thrown back, and so on. And it's sensitive both to external, chemicals that can affect the membrane and therefore change the internal state, but also internal mechanisms that can throw switches. Does that summarize it?
Yes, that's more or less it. The cell itself, we look at this as a 3 component: a component of the cell, and a component of external sensors, which we call the membrane or the effectors, or the receptors, and there are the effector. It's not only the genetic one, it's not only genes, it can be genes in different states. So that would summarize it nicely.
And the specific thing that's exciting about this, of course, is that you can model stem cells, which have the exciting property that they can turn into other kinds of cells.
Yeah, they can differentiate.
So perhaps you could, because this audience in BCBT is mainly interested in neuron cells, perhaps you could fill us in a bit more about the different kinds of stem cells and what we know about them.
Well, it depends. Stem cells in the core of stem cell research is cells that can differentiate and proliferate and carry their own data. But lately, a lot of research was done, and they claimed that stem cells, this naive definition is not enough, and there is a notion of stemness. It's kind of how much stem this cell is, and it means that even in a growth tree, if it can differentiate to 3 different cells, or can get any kind of cell, like the embryo cell, like the zygote. The first cell is the original cell, and this is more or less the new notion of stem cells. But the original one is a cell population that can maintain itself and differentiate. And in almost any organ you can find stem cells.
And even you can, I understand that you can take cells which aren't stem cells, and by stressing them, I think I heard this about blood cells, was it? You could maybe turn them back into stem cells.
Well, I don't like the term turn them back. You can
turn them into
you can turn the stemness mechanism of the cells on. They start to behave like a stem cells.
Right. And so the behaviors of the cells in your model, can you just summarize the things that they can do?
They can do the two most important mechanisms of differentiating. They can go from one state to variety of state. There is kind of a tree of decision that the cell can make. And in parallel, orthogonal component would be to proliferate. They can create new instance of the same cell. And this is enough to maintain a stem cell population. But in our model, there is an additional requirement, and this is their position in space. They can move to another place. This is how we can create structure of organs, and we can create different population. And as we have a front end, we visualize the model. You can see it. The user can view it and can interact with it. Sometimes you can even say, okay, I want cell number 5, 6, and 7 to be erased for the simulation and see what's going on, or to change the environmental effector at runtime. So at the larval stage, or when the model is young, it's not an adult yet, I want to keep the environment as it should be. But At some point, I want to decide that, okay, after 3 days, I want to change the environment. I want to inject a new material. Or you want to kill some of 10% of the cell, and then to see what the simulation suggests the result will be.
Right. So your model consists of a number of initial cells, maybe even just one, and then an environment in which the cell lives, and it's communicating with the environment.
And with each other.
Yeah. So in terms of the movement of the cell, it's able to do things like move up gradients, chemical gradients and so on.
Yeah, it senses the gradients in your environment and can move towards.
So can you just summarize what the environment looks like from the point of view of simulation? So you have chemical gradients in it, for instance. Would you have physical structures beyond that?
Yeah, okay. The environment is divided into voxels, three-dimensional pixels, boxes in the space. And inside there is a gradient of chemicals that exist in each part. In many cases, the decision what are the values is based on the differential equations that determine the value, and it changes over time. So the environment itself is a model of its own. Then in each one of these voxels, there is a cell or sphere that senses what are the chemicals in the atmosphere or in the surrounding environment and act based on that. This concept is inspired by it's a rather old concept that didn't get a lot of attention in science. It's called autonomous agent.
Right.
Autonomous agent is taking out of artificial intelligence. During the 60s, they defined this concept as a machine, an object, or agent that interact with this environment and decide on its next move based on its current state and the environment.
So you consider the whole model to be an agent-based model?
It's agent-based, and it's autonomous agent-based model as adapted to biology. This is like the computer science view of the models. Yeah.
So the first model you described was to replicate the pancreas. And historically, that was your first model, or?
Yeah, that was the first model.
And what was the specific sort of scientific question that led you and the group you're in to think that the pancreas was a good place to go for this?
Well, I wanted to study about the diabetes, and I started research about the diabetes, and then I got to the organ that is controlling the diabetes. And then my view was that you can't study the organ without knowing the process that brought him to this state, and you need to understand the structure and how it was formed. So you need to start by the first day where the organ develops in order to understand what went wrong by the end. And if you notice during the lecture, I haven't even talked about the diabetes,
right? Because
we didn't get this far.
Right.
There were a lot of open questions, a lot of interesting questions that just emerge as we go. But I envision that models like that will cover the whole process, including the functionality in the end. And then you can study diseases and everything. And you can track all the way back to the history and see if something went wrong on day 0 and affected what we see at day 100.
And I guess many of the diseases that you're interested in have a genetic component, which perhaps you'll then be able to recreate in the model once the model is sufficiently complete.
Yes, but it's not only the genetic background. It's like the thing that the organ experienced and caused this disease.
How far does the model have to develop before you'll be able to ask these kinds of questions with it?
It still have right now it covers all the developmental birth. This is 15 days, and we need to add the expression of the factors of the hormones to that, and then we can investigate it. So I believe in a few years' time.
So you're close to having something that would
be useful
to a disease model.
Yeah, but we are close to have kind of a complete model, but it doesn't necessarily mean that we know everything we need. Because once you finished modeling the organ and added all the functionality, you still need to verify that it is consistent with biology.
So at each stage when you're developing the model, you're using some data from the biology to test the functionality. For instance, I think you're showing with the pancreas, you were showing what happens with the interaction with blood vessels and how the model produced similar outputs to what people observed in experiments. That methodology whereby you take these biological data and use them to test the validity of the model. Do you consider that the methodology that you have there is finalized, fully refined? How do you know when you've done enough tests, or how do you know that? Or it's open-ended, I guess.
Yeah, you never have enough tests because the data keeps streaming into your desktop. So there are more papers published, and there is new data, and you have to refine the models as much as you need.
Okay, so, but I guess another way of putting that was, what's the minimal set of sort of biological constraints that you feel you need in order to say that the model is useful?
If you manage to reproduce the biology and a few mutations, so it's not enough to have the biology reproduced. You still need to show that you agree with the known data, and then you can use it for prediction.
Yeah, I guess I'm thinking sort of when people are building a machine learning system, for instance, something that would learn to recognize spoken language and create written language. Then they might take a data set and use half of it for training, half of it for testing. Do you use those kind of methodologies of deliberately leaving out some of the data?
No, I would include all the data,
right,
and then start testing.
But is there a risk then that your model isn't sufficiently well? In order to, you can build the model to fit those data, but then you don't have enough test points to really check whether the model
you should get. I believe that you should get all the available data into your model, then start taking the next step and see if you can get more data once you have the model.
Would that be because you need what data there is to constrain the model and build it?
And I think that the difference is that I'm working with biology. Leaving out some of the biological data makes no sense. So if you know an evidence on the system, you must agree with it. What can be done is that that experiments that were done in the past and you are not aware of, or you prefer to keep them as a test case, that can be done. But for the wild type simulation, you can't leave data out.
So the data set wouldn't be sufficiently rich for this kind of to have two sets of data, one which you used to build the model, one which you could use to test.
In analogy, we can say that you take the wild type data, the type of the normal growth and normal development, and you test the mutation.
Yes, yeah.
So that's a separation you can do,
and that's what you're doing with the models.
That's what I'm doing with the models. But you can take the existing data and say roughly this I keep out and this I keep in. You have to keep something in mind before you do it. And I think this separation of mutation and real data can be equivalent to what you described.
So to come back a bit to the cell model that we're using, and naively you might think about that you would model a cell as, let's say, an autonomous entity with a membrane that sort of is motile, it moves through some substrate, it might have interactions with other cells, adhere to it, and so on. But in the simulation, that's not really what a cell is, right? In the simulation, the cell is in the end defined operationally as the state of a little volume of space, right? So you take the whole volume that you want to simulate, and you sort of cut it up in a lot of little cubes. And then what you call a cell is the state of each of those cubes.
Well, it depends how you... No, I don't like this way of looking. I disagree with it because there are a lot of empty cubes.
Mm-hmm.
What type of cell is that? Is that the null cell?
Mm-hmm.
You can...
Well, tell me, right?
No, I don't believe in the null cell.
Okay,
cell is an existing entity. Mm-hmm. A null cell or a null, null object can be a mathematically defined object, but it can't be a biological defined.
Sure, but in terms of the caricature I gave of the technical approach, the implementation is I think reasonably accurate.
Yes,
in terms of implementation.
The implementation, right? You can look at that. Right? You can look at it that way, but you'll have to add the temporal component into it.
Of course, no.
So
then
it's kind of, you can think of it as a kind of a 3-dimensional space or 2-dimensional space divided into cubes or pixels, and it changes over time. And
so it's like a 3-dimensional cellular automaton.
Yes, you can think about it as a collection of 3-dimensional cellular automaton.
Right, but then
we have chosen to separate between the environment and the cell. And this is because this is how it acts in biology. So it's hard to me to. It's like saying you're not a human being. You are kind of a pixel, and you are just moving from one place to another. But no, that's not the truth. That's not the case. If you're doing something, if we are talking or something, there is something behind this action. We're not just talking. We're having conversation. You say something, I reply. So to think about it just as movement and things in the space, I think it will be simplification of reality. But yes, if you prefer to see the technology challenge the implementation,
just to define what we're doing, right? Because we're interpreting the outcome of an algorithm, right? And the algorithm is just, if you want, deciding what state a certain voxel in that simulation should take, a certain little bit of this 3-dimensional volume, and then with that you model these systems, these organs or C. elegans and the brain. These are the 3 examples we looked at. So then the question becomes: okay, when is then such a simulation, let's say, defendable? When is it plausible? When is it not more our interpretation as opposed to what's really going on in the simulation, right? So for the pancreas, you showed us that we have to look at a system that essentially at the stage in development you're modeling expresses 3 cell types, right? And these three cell types cluster in very specific forms, no? And then what you want to recover in your simulation is, again, the generation of these 3 cell types and this kind of clustering. Is that correct?
Yes, that's correct.
Okay. And then I could argue, well, and then the structuring of this pancreas was then also very much predicated of the role, as you told us, on the role of the blood vessels, right? So the blood vessels providing some sort of scaffold in which that very much guides the coagulation of the cells. Yeah. So, but then this
is a well-known biological fact. It's not something that we
no. But you could still argue the biologists were not really able to tell you yet how this then happened in all its details, right? How sort of, let's say, specific parameters might affect this. But then you could, as we saw earlier, right? Your cells, as you said, have dozens, hundreds of states. Large populations of them give rise to specific structures. But what we try to explain are the 3 cell types of the pancreas forming, like cauliflower type structures. So I could say, well, this is a super powerful model. You have so many parameters you can play with. This can never go wrong. You should always hit the jackpot with that, right?
Yes, but all the parameters in the model are constrained by biology. So I believe that what is in the model, it's in the biology. So maybe the biology is superpower model. You can think about it. It's right, because you can find the pathways in cellular decision that you can bypass. You have a few pathways that leads to the same result. So let's ignore the model and think about the biology. Why the biology have 2 paths that goes to the same destination?
So another way of validating the model is to create variants of it which don't fit the biological data. So just take the one which you think is accurate and flip a few of the arrows or whatever and see if they do anything similar. And that would tell you how valid it is to say that specific model is needed to generate the data that you see. Now, in some sense, that's what you're doing when you get your mutations, but then you're saying this is an actual mutation that happens in nature, and I can see what happens in the model. But have you done experiments where you just flip components or leave components out and see how that affects it?
Yes, of course. One of the first experiment in silico experiment we did was just to take the nucleus, the inner decision of the cell, and just make a big spaghetti, like Erdos way of testing, taking the arrows and connect them randomly to other states and to see what's going on. Like the craziest stuff. It's just like taking the DNA and just take the operon and point it to express a different gene, just like a crazy scientist do. And yes, this disabled everything in the simulation, but at least it's proven that that data is really necessary for the modeling. But what I'm trying to say, and maybe I'm not clear enough, we are trying that the models will be realistic as possible, and to take off a pathway or take off genes just because we don't understand what they do, or there are other genes that do the same, that does the same. I wouldn't recommend that, because then you might miss point in the model. The idea is to put into the model as many biological data as possible, not the minimal one.
Well, but there are two issues for me on this, right? On the one hand, how do you really quantify a cauliflower, right? So in some sense, in the presentation, it was a bit more sort of by eye, like, well, so it looks sort of similar, right? And I think we would all agree that we can do better than that, right? And also on top of that, cauliflowers have fractal-like structures, which raises interesting questions about development, right, and morphogenesis. So how then do you really quantify that similarity between the structure that you see in your simulation and that you find in biology?
It's not easy. I haven't.
Life is hard.
Life is hard. Yeah, and one of the hurdles in this issue is that there are not that many biological data. It's not that I can ask a biologist, please give me how many cells there are in this cauliflower and what are the fractals, and this will be a very naive way to see the experiments. Usually that's what we get. That's the data.
Still in the literature, you will find these characterized. There will be images, there will be microscopic analysis, electron microscopy, right? There will be different levels of analysis.
You will hardly find it in mice. You will find it in, I think, I didn't come across too many.
Or, for instance, you might have the inter-cauliflower spacing.
Yes, what I did, for example, is doing some of image analysis, trying to compare not exactly the population, but to split between the color of the domains of the color. How many pixels carry red color and how many green colored pixels there are in histology picture, and to compare it with the cell count in the simulation. So it's kind of making...
Okay, but you're saying you couldn't really do that yet because the data wasn't there.
The data is not there.
But
the data is very dirty in this.
Okay, but which parameter in your model would, for instance, control the intercauliflower spacing?
There is no single parameter. Okay. There are a few parameters, and all related to the biology. For example, the flow of the gradient of the gradient of the blood vessels secreting the factors in the environment. That would be a very important parameter. And indeed, when we played with this parameter, we see that we generated a lot of different structures that may and may not be correct, but this is something to be tested in the future when we can control the blood vessel, the way the blood vessel secretes factors to the environment.
Right. So then, after the pancreas, we looked at the C. elegans, right? And there in particular, you looked at the hermaphrodites who are basically producing other C. elegans by self-fertilization. And that C. elegans, the way you described it to us, works a bit like a sausage machine, right? So you have sort of stem cells generating the cells that move through the body. They then sort of get transformed into eggs or a new stage, and then they pass through a sperm phase or they got merged with sperm, and then there you have
they get
your
fertilized.
Then you have your fertilized
but
egg,
it's
right?
not. It is big part of the C. elegans, this reproductive organ.
Sure,
but it's not all of it. It still have a neuron system and locomotive system.
Right. But you sort of you modeled this sausage machine that sort of is spitting out, if you want, these new C. elegans.
This is the gonad. This is the reproductive organ of the C. elegans.
And so, which elements of the simulation of the pancreas could you carry over to this C. elegans system?
Well, by means of platform, I used the same platform.
Principles more.
And the same principle was implemented.
Which is?
And the way we construct the cell and the cell different components, the nucleus and the membrane and all the autonomous agent concept we discussed earlier was just adapted to this model. We had to change the name of the genes. But this is basically it, and the connections
but
between the states.
All the blocks are different. Yeah.
And the way they affect each other. So if we think about it as a graph, we just had a different graph between nodes and arrows.
But I would assume that also the number of states of the cells would be changed.
Yes, right? Yes. If in the pancreas we had like 120 states, in the C. elegans we had like 20, which is like five times less.
So in the environment in which the C. elegans is growing, what is that environment? How, because the pancreas is growing in a very different kind of environment from a...
Yes. For example, in the pancreas model, we had blood vessel, very complicated network of blood vessels. In the C. elegans, it's very easy. At the tip cell, there is one cell that secretes a factor to the environment. That's it. So it was much easier to get a more accurate description of it, and it was even modeled as using ordinary differential equation at the stationary phase. So it's just like a chemical that is being secreted in a gradient.
Right.
It is much easier to capture. So it's like one cell of the blood vessels of the pancreas model. Yes, it's much easier, and this is why it was much easier to generate good prediction or testable predictions. While in the pancreas, every prediction I had to wait like 5 years until the biologists find the right technique to test it. In C. elegans, it was tested within weeks.
But now in the pancreas, I think the benchmark was very much the morphology of the structure. So it's correct, right? So what would be that benchmark for C. elegans?
I think it's okay. It's more accuracy, because most of if in the pancreas model, I had to use a lot of hand waving and say, okay, look, this is similar. In the C. elegans model, I can talk exactly about the lengths of areas and the lengths of zone and number of cells and cell cycle. And indeed, out of all this refinement, fine tuning of the model, it turns out, for example, even in the small details, what is the different cell cycle between the adult and the young C. elegans during development? What is the difference? What is the ratio between the cell cycle at the two phases? And we suggested in the model 1 to 5, and then biologists think it's 1 to 3.5. So it's at the same range, and we didn't take it into account. We just try to make it as realistic as possible and to calibrate it with time. And this all tiny detail just emerged.
So that would mean in for C. elegans, the benchmark is more like what kind of cells do you have in C. elegans and what position at what point in time?
And the number of cells.
Sure, yeah,
the number
exactly.
of cells. So you can start talking about numbers and quantity aspects, while in the pancreas it will just look, it looks like a cauliflower.
Yeah.
It's similar.
But in for C. elegans, we talk in the end about 3 or 4 cell types, that the stages, right? So why is that a hard problem?
Why this is a hard problem? Because it's not all about the not about the cell type. What controls them? What is the network? This is harder. How the external signal affects the development. That's a hard problem. It's hard to know what happens there. And not all is known. Many issues that are open. For example, what causes cell death? Is it autonomous? Is it decision? Is there an external signal that kills them? We know that the 30% of the cell do not make it to the 30%. It's a lot of them. It's like third. So we know that they don't make it. What happened?
Yeah, but I found this interesting, right? You mentioned this also in your talk, that this was one of the insights that you had. But apoptosis of cells, so the cell death, is an active process, right? This is really a regulated process. There are receptors sitting at the outside of cells that are directly driving apoptosis. So but it's not like, well, in your simulation, you were telling us you were inducing cell death, but you're saying, okay, if cells pass through this zone, there's a 30% chance they won't come out.
That's because apoptosis is very active in the adult. In the developing organ, it's less active. Usually, how many cells an adult is losing in his lifetime. The embryo keep growing. Usually, developing systems has less apoptosis than the adult. So,
well, they better, otherwise they won't develop, right? No, what I'm trying to get to here is that in C. elegans, we would assume that also their apoptosis is actively regulated. For instance, dependent on environmental factors, apoptosis might be more or less, right? Depending on, let's say, the stress on the animal. Well, in those elements, you are not really including in your simulation.
No, we left it out. We just flipped a coin at that
exactly.
stage. Yeah. This is an extension to the model. I'm not sure there is enough data about apoptosis in C. elegans at this stage. I'm not sure about it. I don't want to commit. But if there is, it can be formulated and added to the model.
Okay.
I'm still trying to understand how you build and tune these models. So in the case of C. elegans, you have what you described as a very simple environment with a chemical gradient. And then you have your cell model for your initial stem cell, which presumably when you set it up, has some of the relevant details from the biology, but not all. And then I imagine you drive the model, you see what happens, and you see, well, it's not building a worm for me yet. And then you say, okay, what else what's wrong with this? What can I do different?
You go back to the library and read many more papers.
So it's a very iterative process where you run models, and all the time the models aren't working, and gradually they get closer and closer to what you would like to see.
Yes.
Okay. And at that point, you say, well, I think I've got a reasonably good working model of the wild type worm. Now, then you go to a different part of the library and say, let's look at mutants and say, let's see what happens if we now flip the circuit. Do we get mutants? Exactly. So the validation stage is it's quite a strong one, really, because I think you already have something that captures the development of the living natural animal, and then you can mutate it in ways that you can be fairly confident match real mutations.
Yeah, but that's a very important part of the modeling process. But once you complete it, this is the really interesting part. You start to play with it. You start to risk it. You can play the crazy scientist. You can do whatever you have in your mind, and then you can get predictions. And some of them are testable. And for some of them, you can tell whether they are plausible and whether they are testable. Some of them are just too crazy to be tested.
Some of the predictions you make from the model turn out to be wrong.
Yeah.
And then
very often. Then you go to stage one, go back to the library, see if
so the methodology doesn't change that much. I guess you get your basic working model, you try it out with some mutants, some it works with, some it doesn't. You say, well, it didn't work with that mutant. I need to refine the model again.
Yes.
Yeah.
But no, there's
there are
no
version of models.
Yeah.
All the time. And this is a never-ending process because data flows in, and as the data came to your bench, you just need to rethink all the model. Sometimes a small paper can change the whole model.
But what I find interesting is that apparently you live in in a field of developmental biology where the literature is pretty clear, because apparently you can just go to the library and pick out the papers and get the parameters for your simulation. And in reality, at least the field in which we exist, it's never like that, right? You find the papers, they're contradictory, they're incomplete, you have to meditate on this stuff. They have to talk to the specialist to actually understand what the hell they're writing about, right? So is it really fair to just say, okay, then I go to the library, get the papers, I know my parameters? I cannot believe that's how it works. Right.
That's not fair
exactly.
at all.
So how does it really happen?
Every 3 papers are 4 different options.
Exactly right, yeah.
But the modeling approach allows you to change between hypotheses and try to check different cellular mechanisms.
So you can play out scenarios, right? That's the whole point, right?
That's the whole point of modeling in general, I think. But
I agree.
What the modeling, my modeling or the modeling I presented offer is kind of a nicer visuals way for developmental systems, in particular at the population level. It's not a single pathway that we are testing. It's not one cell. It's the whole population, and it's the whole population over time and space. So you can get a lot of new insight and ask them all new. A lot of new questions.
So that means also the model can summarize a lot of data. It can help you to play out different scenarios to see, okay, what if all this missing information would have this characteristic?
It could also highlight what you don't know, because it summarizes it.
Sure. But now, in the end, if you want to turn that model into a theory, you must explain something and you must make predictions, right? So at this stage, if we look now at the model of C. elegans, what do you think have you really explained, and what is a testable prediction of that?
Yeah, we explained a lot of the interplay between cell cycle and differentiation. And one of the insights I presented is that if you change the ratio of cell cycle between the larva and the adult, then you get different behaviors. And one of them is that your stem cell population gets shorter or smaller once you reduce it. It seems a bit naive and it seems a bit straightforward, but the scientific community never thought of looking at the consequence of the cell cycle at the larva stage on the adult stage. So this is kind of question that we can easily answer or ask. What happens if you change something at day 0? What happens in day 5?
Right.
And it's not that easy to test in the lab, because you change something and you need to wait a few days. It's better to have kind of where to look at, not what the answer is, but where to look and search and research in the system,
right?
And that may give us an answer about it.
So then the last example you analyzed and presented was neurogenesis, right? The development of the nervous system. And again, you roughly took the same modeling framework, the same cell model, but now again changing the states, changing the expression pathway or the control pathways to build a piece of brain. Right. So what were the insights there? It's a piece of brain.
It's a piece of the neurons and the nerve system, because what you see there is just what will be later the nerves.
So what did you exactly learn from that exercise? What did you find?
Well, before we go there, can we, in terms of actually building that model from the previous 2 models, were you able to take little networks of the blocks from within those models and use them again? Because we're interested this week in how evolution has reused some basic processes, perhaps in very different ways, sort of regulatory gene networks that might be involved in body patterning and invertebrate reused again in building bits of brain. And I wonder if, there are examples of that in your work, that you can say there was a network in one part of my cell and I was just able to use it in the pancreas, in the C. elegans, and in the mouse brain.
Yeah. First, I'd like to give you some statistics that may explain it. The first model, the pancreas, it took 5 years to develop. The second model, the C. elegans, it took 2 years, and the model of the brain, of the neuronal migration, took 3 months. So we short the time of developing the model itself. The analysis keeps taking long time, and looking for the prediction is a long period. But to develop, since we have a good view of what the modeling framework and the approach is, it takes less time to develop the model. So the basic concept and the basic approach, as time goes, as the research continues, it's much easier to implement it. And this concept of autonomous agent and the concept of having this sensor unit and the internal switch unit and the differentiation and proliferation are being controlled by these two components seems to work. So this seems like a general concept in developmental biology. I'm very, very careful here because I don't want to claim, hey, I have the holy grail, I'm having the way to model, but it seems very beneficial. We are encouraged by the results. It doesn't mean we hit the jackpot, it's kind of, we still have to do a lot.
But what was the benchmark? What was the benchmark in this case? What did you replicate, and how accurate was that replication?
In the neuronal migration, we replicate the way neurons migrate from their birthplace to the place where they are going to become part of the nerve system. So it's from the core of the brain to the surface. It is guided by fibers, but the glial fibers. And at the first stage, they use the glial fiber to track the road. Then something happened, and they stop following this glial fiber, moving randomly in space. Then they reattach to the glial fiber and just putting layer on top of the layer of neurons on the surface of the brain.
But now, so in your what we're really talking about is also really the migration of cells along these guiding axes of glia, or certain kinds of gradients that might attract them or repel them. So that means in that setup, collisions is an issue, right? That cells, if these migration patterns get disrupted, cells might not be able to migrate across certain other cell population.
In all cases, they do not migrate one towards the other, one across the other. If they see that there is a cell in a neighboring pixel, they won't move to it.
No, wait, but in reality, cells have to go through layers of cells that might have embedded themselves earlier, right? Yeah, because you always go to the outside.
Yeah, but you need to think, okay, it's not exactly three-dimensional, not exactly two-dimensional. It's somewhere in the middle. So there is a layer of cells. They climb above it. They're creating another layer, and they are walking on top of them, and then they will place themselves on the same layer. So it's kind of two slices. So they go over and.
No, but what I was trying to get, Zoltan Molniar was here early this week talking also about development of the brain, and a typical pattern there is that you have certain subpopulations moving from these neural plates at different points in time, and also having to cross through.
They cross through others,
right? And they cross through other populations. And what I wanted to ask you about is that my claim would be that with your simulation technique, you cannot handle that, because in one location in space has just one specific state, right? So the dynamics of, let's say, migration and possible collision and obstruction and so on is not something that you can capture that way, or would you think that's a too negative interpretation?
Well, I think you've just defined a research question.
Okay.
It's enough to be a PhD thesis. This is something to investigate.
So I might have a chance to get my PhD done.
At my lab, yeah. You're accepted.
Okay, great.
But you still didn't fully answer my question, and maybe, I don't know if it was a bad question, but you said that for the brain model, you keep the same structure of the previous two models for the cell. But what I was interested in, all these cells are doing similar behaviors of differentiating and migrating. Are there some bits of network within the different decision-making parts of the cell that you could reuse from one cell type?
The proliferation, the cell cycle is the same through all the models. there is the decision about asymmetric and symmetric proliferation, which is different between them. But the proliferation component is the same, and the differentiation component is very similar.
So this is exciting in terms of when we think about evolution, and we think, well, brain neurons are very different from C. elegans neurons, but actually they share a lot of the same chemical machinery for doing what they do, and you have to change the specificities of what they're doing, but same networks can work internally.
Yeah, I think this is the discussion that we had at the end of the talk, that you think that you're surprised that the biology has simple building blocks, and I see it as the reality. It's kind of, it's surprising that the biology carries simple building blocks, but I think this is the way it is. I don't think it is as complicated as we'd like to.
Not wanting to agree with either of you, I could argue, you guys are both deeply confused, because
we're scientists. Of course we're deeply confused.
So, right. So, but the point is that in your case, what carried over between these phenomena was a modeling strategy, an algorithm, if you want. But that technology as such doesn't tell you much about guiding principles, because as you said yourself, you don't have all these principles explicitly defined. And Tony is seeking, let's say, common principles across all these different things. Like the way you would grow an organ, a pancreas would include principles that you could carry over to having a C. elegans sausage machine.
But I think we said that there were some intrinsic cellular networks that you could copy whole chunks of the network across from these two different cell types.
If you prefer to think of it as a mechanism motifs, that this is a motif of a mechanism of a cell, this is another way to look at it.
Because, but then we have to, if you gentlemen are so optimistic about this, you must declare from
we're scientists, we must be optimistic.
Right here and right now, what the common principles are between pancreas, C. elegans, and this piece of brain.
So they both have proliferation, differentiation. Okay. They both have a kind of component of external sensor recognition or signaling, and they both have kind of internal inner cellular decision. And the decision of the mechanism must be defined by all the three components. Okay, it will be wrong to say that the environment and the extracellular signaling is not important, and it will be even more wrong to say that the internal genes is not effective, and it will be wrong to say that this cell has no proliferation or no differentiation. So this is necessary components for all the three elements, maybe whatever it is, whether it is the pancreas in mice or the C. elegans germline development or the neural development.
But the risk is, of course, that you say, look, E. coli can have flagella and they can sort of flop around in some gradient, and humans walk and they share common principles because they navigate. So the point here is that, for instance, I could argue that in case of the pancreas and the brain, we might have also the regulatory genes that really tightly orchestrate this process, while the same might not hold for the complete pathway of the C. elegans or the trajectory that cells go through in C. elegans. You will not have single regulatory genes guiding and orchestrating that process.
Well, it's never single gene. It's kind of combination of many genes. So it's between one and many. But with the bacteria, with the E. coli flagella, it's a different system. It's not developing. It's hardly proliferate. If you want to see it,
I meant something else with this. It was like seeing a similarity between the flagella of E. coli in our legs, because they're both used for navigation, and say, this is now possible to have a common principle.
Maybe they are. Why do you rule it out?
No,
but I mean,
seems a long shot, though.
So those abilities are probably in our genome. They're just not expressed. And I think what you're saying is it's not surprising, because we're all descended from a common ancestor, and C. elegans and the mouse and ourselves. In that common ancestor, there were stem cells. They migrated. They built complex bodies, and so what more do you need than those mechanisms? So, so most of it's already going to be there. And you say you're not surprised. I'm a little bit surprised, because I think that maybe in rodent brains, there's more that stem cells do, but for you, that's not a qualitative shift in what they do. It's just a bit more richness, perhaps.
Yeah, it's more richness. I don't think it's more complex.
Yeah.
It's kind of...
So look, now that you gentlemen refuse to see the light I'm shining on this issue, maybe we should get to the finish line. And the question there is, so Yaki, as you also shared with us, it's actually interesting, the community of people doing your kind of work, like modeling these developmental, is actually rather small.
It's very small. Yeah.
So you're sort of chipping away at this, making progress. But in that sense, given what you've seen and learned in this sort of, as a solo agent in this field of developmental biology, what would be Yaki's law that we should adhere to in trying to understand the developing biological system?
Well, keep it dynamic. Look at the dynamics.
Okay.
I think that is the most important. I think many people in this field fail to think dynamic, to think that an event now has effect in the future of the animal. And if you look at that, you'll find an all new world of results. That would be my law.
Okay. Now, Tony likes traveling, and so I ship him around the world.
So when are you coming to Israel?
Exactly. I'm going to tell you that. I can tell you this exactly. 4 years from now, he's going to come to Israel, and he's going to check you. He's going to meet up with you, and he's going to check a prediction you're going to make today. Tony's going to ask you, look, 4 years ago, you made a specific prediction that you would observe X in your simulation. And today I want to know, 4 years in the future from now, whether this was validated. So what's the one prediction you're gonna make for us today that you will see tested by then?
Yeah, well, I'll give a different kind of prediction. I'll predict that more people will do my kind of modeling.
Come on, you can do better than that. It has to be about developmental biology.
Developmental biology. Okay, so I predict that more people in development biology will do. No, I really think that what I'd like to see is not prediction in the lab that I've predicted and was accurate. I would love to see it comes out of the model, but I would love more to see people doing my kind of science. And maybe in the long run, there will be a model of a human being that we can play before we go do anything. So that can be something. This would be my vision. I'm not into looking into a particular prediction that will be validated.
All right. Yaki Setty, thank you very much for this conversation.
You're welcome.
And Tony, you're a bastard. You're supposed to make it difficult for him, and you're not supposed to agree with him.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.